if they're gonna feel like we can be back in the fall.
Wow.
Yeah, I mean, it's too early to say, but
Right.
it's a discussion point.
Okay. Yikes.
Yeah, yeah. So,
And our commencement will get pushed back even further than August.
Oh
well,
my goodness.
yeah, yeah. I mean, you know, some schools are just going with the route of like, let's just do a virtual one, like in May, you know. Um, and I don't know that, I don't know, pros and cons of like, trying to push it off and I guess actually I read one university was doing a virtual one in May, but then also going to invite 2020 graduates to come back if they want to like join in with a later one. Maybe that's kind of like a good splitting the difference. I don't know. What do you guys think about
Well, that's
graduation?
what Ferris is doing.
Is it?
Yeah, Ferris is like having a virtual one in May, and then if you want to come back for December commencements, you can, but I think it's kind of like that would be way too much for people to do.
Like December's too far off?
Yeah, I mean, because like then you also have like the graduating class of that fall too.
Oh yeah.
Like,
Yeah, that's true. Good morning, everyone. We were just chit-chatting about life and I was checking in with stress levels, seeing where everybody's at. Anybody feeling super stressed? Feel free to unmic and talk. I was saying I feel like I miss hearing your voices versus, like just typing. You can type if you want, but you could unmute yourself too. Anybody feeling super stressful?
I don't think it's the stress. I think it's just I'm still in the adjustment phase. And I'm the kind of person that I feel like I have to get everything done the minute it's assigned. And so I've been like picking like 3 days where I just work for like 16 hours. And then the rest of the days I don't know what to do with myself because all of my assignments are done.
Ah,
So I got to figure out a way to divvy it up a little better, but we're getting there.
yeah. I think probably each one of you is probably doing things a little bit differently if I had to guess.
Mine's
I feel
definitely
like I feel like the drafts of the papers are coming along really well. So we're almost there with those. One last section. And then, making the revisions hopefully isn't too bad. Like, right? I mean, I think the harder part is getting the first draft done. What do you guys think? What's harder, first drafts or revisions?
Definitely. I
Yeah.
feel like that's the majority of the work right there.
Yeah. Finding the papers, reading the papers. Yeah, I think so too. Hopefully the revisions won't be too bad. Okay, I was saying I got on a little bit early today to try to experiment with sharing my screen because that was such a disaster on Tuesday. It's still not working at all. Webex just freezes and crashes, and so I'm not going to share my screen and it's so strange. I mean, a week ago I could do it no problem. So I'm not sure what's going on there. I don't know if the system is just like really overloaded, like Tuesdays and Thursdays are just really they are popular class times, or I don't know if it's something with my laptop, not sure. But I don't want it to crash because those were like some big lags on Tuesday. So I have all of those documents like pulled up. So if you have them pulled up and you have questions about anything, we can kind of go through them one by one and we can see if there's questions, or if you just want me, even if you don't have a question, I know sometimes it's hard to come up with a question, but if you want me to explain something, like you're not quite sure what something means, I could talk through it and maybe that would help. Um, so let's maybe do that. And if you have a suggestion, I'm happy to take them. Just want to try to make this like to be a useful session. So we could start maybe with today's assignment. It looked like most people got that done. Today's assignment was I'm pulling it up. Sorry, I thought I had everything pulled up, but I did not have that one pulled up. The 460 assignment. Let's see. There we go. Okay, so there was the PowerPoint, there was an animation that explained microarrays, and then there was the article. So there were those 7 questions. Did anybody have any issues finding answers? Was anything unclear? Do you want me to say some basic things about microarrays? Let me know. Let me know what's helpful.
I think it was pretty straightforward. The only thing I was kind of unsure about was some other things like that are the question 2 basically. That could potentially do the same thing as microarrays.
Yeah, let's talk about that.
I think for the most part, the guide was really straightforward.
Okay. Okay. Well, I'm glad it seemed like it made sense. Okay, so in order to answer number 2, we need to make sure we're clear on number 1, which is talking about how microarrays can distinguish whether gene expression changes between 2 or more samples. And so the first question was, what level of gene expression are we looking at? And the answer is RNA. So that's what we're looking at. And then question 2 says, what's another technique that could also give us answers about changes in gene expression at the level of RNA, but not necessarily at a genomic level? And so my first answer, and I think it's in, let me just double check. It's in the PowerPoint for today, the 414 PowerPoint on slide 3. It shows you a northern blot. So that would be one method that also gives us information as to whether or not RNA changes between 2 samples. So on that picture, or like I drew a little cartoon of a northern blot, you know, we've drawn those a couple times during the semester. So in the control, maybe you would see a nice band showing that RNA is expressed in your control. And it's possible in your experimental sample, whatever it is you're comparing, that there's no band, right? So that would tell you that RNA is being transcribed in one sample, the control, but not in the experimental. It's not always that black and white. It could be that there's a nice bright band in the control and in a fainter band in experimental, right? That would tell you they're not being expressed in the same levels. And that's a big thing with microarrays, is getting the levels of expression. And you can get that from a northern blot too. The big difference between those 2 techniques is that the northern blot's giving you information for one gene at a time. When you do that blot, you're running the RNA on the gel and you're using one probe for one gene, right? So you're getting information about that one RNA. In contrast, the microarray, you're getting information for thousands and thousands of RNAs, whether, you know, what levels they're being expressed at. There are other answers besides northern blot. I mean, you could talk about RT-PCR. Did anybody have any others that they came up with? Did that help, Hannah?
Yeah, it did. I think the wording that threw me off was the genomic level, and I was like, does it have to be the genomic level or not?
Yeah,
What
I
you're
think
looking
that's,
for.
yeah, sure, absolutely. So, I'm sorry, I have like a lot of files open, so I'm just going back to the wording of the question. Right, so I said not necessarily at a genomic level. Well, certainly northern blot's definitely not nearly at a genomic level.
Right.
It's right, sort of 2 ends of the, you know, of the spectrum. And I don't know if you guys have noticed, by and large, I've been grading these assignments just on completeness, especially these last couple. So don't worry if you didn't get something exactly right. As long as you made a stab at it and it looks like you know, gave it a try and came up with something semi reasonable, it's all good. Any other questions on today's assignment about microarrays? Or any follow-up questions on what I was just talking about? Oh, figure 3A, sure. Okay. Yeah, so that's a great one to go over, Oliver. And this is where I really wish I could share my screen, but I can't, so I won't. I will resist that temptation. Figure 3A is from that Stout paper. It's in the PowerPoint. So just so you, if you want to make a note, what I'm describing is slide 14 in the 414 PowerPoint, okay? And what is on this slide is figure 3A, which is titled diffuse large B-cell lymphoma. And there's a picture like histology showing us some cells. And basically, right, that this is some sort of cancerous, you know, type of cell. I'm not very good at reading histology, so I don't know what that means. But the microarray below it, I can walk you through. So this microarray is what you would get after the software looks for patterns, right? So it goes through that hierarchical clustering to look for patterns. It's looking for patterns in the genes. It's also looking for patterns in the different tumors. So just as a reminder, for every microarray, every row is a gene and every column is a tumor. And if you kind of like, this is a lot of data in this one little figure. So there's lots and lots of samples here. Like it's hard to blow up, you lose the resolution, but there's a ton of different genes, like those little squares, they're tiny. You can kind of see the black ones. They're really small, right? So that kind of gives you an idea of just how many genes there are, like how many rows there are and how many columns, if you can like actually kind of look at the little black square. So there's a whole lot of them. And the point is that when people were diagnosing patients with diffuse large B-cell lymphoma, they saw differences. Some patients did really well and lived, 5-10 years, and other patients did not do well at all. And so they saw like this big variation in survival, and it made it really hard to tell someone upon diagnosis, like what's the prognosis, right? And that makes a big difference to patients. They also probably saw that some patients responded to some treatments pretty well, but not all patients. So this kind of gave them the idea that maybe it's not just one disease, right? If you have patients who are surviving such different numbers of years and responding to treatments in such different ways, maybe it's a good idea to dig into this. So what they did is they looked at a whole bunch of samples from lymphoma. So every column is a different lymphoma patient. And then they looked at tons and tons of genes. And they ended up finding these patterns, which suggested there are 3 subtypes of this type of lymphoma, right? Germinal center B cell-like on the left, type 3 in the middle, and the activated B cell-like. And the reason they were able to come up with 3 subtypes is because of the differences in the gene expression. So on the one on the far left, it was red on the top and green on the bottom. So all of those genes in the top rows are being upregulated. They're being expressed at high levels, and all the genes in the bottom rows are at low levels. Okay, so that was characteristic of all of the different people, all of that group of people. And so they gave that, I don't know why they called it what they did, but they gave it that name, germinal center B cell-like. Okay, and then in the middle was type 3. So for that group of patients, none of the genes were upregulated. They were all at pretty low levels. And in the third group, it was kind of the flip of the first group. Okay, so in that third group, the top rows of genes were at low levels and the bottom rows of genes were at high levels. So it was green on top, red on the bottom, right? So that was the flip of the first group. And they named that third group the activated B cell-like. So they came up with the groups of types of lymphoma based on how many patterns of gene expression they saw. And then they could also kind of group the genes, right? So they had all in the top row, there were all these genes that seemed to be regulated similarly. And then the bottom were groups of genes that were regulated similarly. So that's how they came up with these 3 subsets or 3 subgroups of lymphoma. And at the bottom of that figure is the survival. Those are called Kaplan-Meier survival curves. And what they saw is that if you separated out those patients based on what the microarray said were the subtypes, that you could actually predict the differences in survival. So if you have germinal center B cell-like if your gene expression matches that germinal center B cell-like, your survival probability is much better, right? It's like 50% to live 10 years, which is much higher than if you have the other 2 kinds. So the clinical data, the survival curves really kind of back up that there is a real difference. Like it matters that your genes are expressed differently. It's actually going to affect how long you live. Okay, questions about what I said? Hopefully you were able to look at that at the same time I was describing it. Follow-up questions? Okay, good, good. Glad that was helpful. Any other questions from that PowerPoint or the assignment? not seeing anymore. So let me move on to asking about kind of part 6 that you're going to be working on, the last section of your paper. So the last section of your paper is this well, you're doing it in an outline form, so that's a little bit different. And you're going to come up with 4 experiments for undergrads, 5 if you're a grad student. And there are 3 different kinds of questions, like different types of experiments. It could be an experiment that is meant to improve diagnosis. It could be to improve treatment, or it could be to better understand the mechanism of gene action. So what we mean is how the protein functions and or how the mutant protein functions, right? So you're going to have 4 different experiments from at least 2 of the categories for undergrads, and grad students are going to have 5 experiments, and they need at least 1 experiment in each of the 3 areas. So, so the outline hopefully is somewhat straightforward. There's going to be 5 pieces, A through E. And if you could write this up like in that outline format, like just keep it as an outline, you can just type right into the outline. In your topic sentence, you're going to let me know if this is an experiment for diagnosis, treatment, or understanding gene function or molecular pathology. And then second, you're going to give me the rationale for the experiment. Why is it a good idea? And then part C is the method and relevant controls. If you describe a method we've talked about in class, you don't need to have a reference. You don't need to cite a reference or a source for that method because we all know them. If it's a method that we have not discussed in class, please do include a method, a source for your method, right? And then, so you've got that proposal of what it is that you think should be done in this experiment. And keep in mind, it's an experiment that you would actually do in lab. So it's not something that you would typically do directly on people. Okay, we can talk more about that. And then part D, you're going to describe the possible outcomes of the experiment, right? What are the possible results? It might work one way or it might work some other way. And then lastly, you'll interpret each of those outcomes. So if I see whatever blank, then I will conclude blank. Okay, so that's the interpretation. So that's what you're going to do for the outline. The outline is due Sunday at 5 o'clock. So you still have a couple days. If you get totally stuck with this, or you just want to run an idea by me, go ahead and email. Feel free to email, okay? I'm happy to give you some feedback before you turn in the outline. But of course, the whole point of the outline and it being a revision is that I'm going to give everybody feedback. I'm giving lots of feedback, as you probably have seen so far. Okay, so with that outline in mind, then there's also those tip sheets. There are 3 different documents in the research overview folder. How to design a gene therapy experiment, how to design a CRISPR experiment, and how to design a microarray experiment. And so that just kind of walks you through thinking about whether the method is like whether it makes sense for answering your question. And then the second part for each of those documents is telling you what kind of information to include in the method. And then the possible outcomes, like what, just reminding you to tell me what it is you're going to measure. What are you going to assay? How are you going to know if it works, right? Are you looking at a phenotype? Are you going to measure, amounts of protein, or, what is it you're going to do? Okay, so hopefully you've had a chance to look at those 3 documents. If you haven't, you could pull them up right now and you could take a look at them and see if they make sense. Questions about part 6, questions of just this whole section of the paper, brainstorming, thinking about experiments to propose.
So, Dr. Skiza, for a lot of these, we're thinking that they're laboratory experiments. What if we wanted to do something like a GWAS, to kind of come up with associations, even though that's not quite lab work?
No, that's fine. Yeah, so that's a great question. So, yeah, doing a genome-wide association study, I mean, is perfectly reasonable. Yeah, and one thing I would say is, it doesn't have to. I want to come back to that idea of like you don't want to propose an experiment like on humans, right? So what I would say is anything invasive. You like if you want to pilot test some treatment, you're not going to jump to doing that on humans initially, right? You don't like move into clinical trials. First, you're going to do this on some kind of model animal. So mice are commonly used, primates sometimes, but there's less and less of that kind of work being done. So the mouse or the rat, rodents are really common systems for trying some different treatments. So that kind of like invasive or, you know, potentially risky kind of experiment, that's where you should avoid the humans. However, when we think about these microarray experiments, for example, and then this would be sort of similar with genome-wide association studies, um, you know, getting like a sample of a tumor and then doing some analysis of it, that's perfectly fine, right? That's actually using the sample from the patients, perfectly fine. So, you know, similarly, like the genome-wide association study where you're doing, you know, looking at a whole bunch of SNPs um, from people, that's also fine.
Okay.
You just want to kind of keep in mind, like what are the guidelines overall that that it seems reasonable, you know. So, like for example, and this is not exactly your question, but I'm just thinking of things that sometimes come up. Um, you know, like the skin, our skin is really like you could take a skin biopsy pretty easily, and that's like not super, um, I don't know, like it's not super invasive. I don't know, I'm trying to think of a different term, but you know, right? A skin biopsy is okay. Like people have skin biopsies done all the time. It's not really super risky. Like you're going to be fine after a skin biopsy. Um, However, like, so, you know, if you have a disorder that affects the skin, that would be something that would be easy to kind of access. In contrast, the brain tissue is not accessible, right? So getting a little brain biopsy is not something that you're going to do on any sort of routine basis, just as like a contrast. So you just want to kind of think about that aspect as well.
Okay. Thank you.
Sure. Any follow-up thoughts, like, or questions? I'm going to say one thing I said on Tuesday, and I think I'll type this up and put it into the Blackboard announcements. I'll do that today. Is um, don't look at the literature, just to remind you of that. Um, it turns out to actually be a problem because a lot of experiments have already been done, right? Some of the experiments that you would think of, and then you look and you're like, oh gosh, it's already been done. And you're going to maybe even, well, you've already read a lot of papers at this point. So you know a lot of experiments have been done. So if you kind of go look, like, has someone ever done a 2 hybrid to see what proteins interact with my protein of interest? Chances are you might find a paper. Don't look at it. Pretend you don't know that, okay? At this point, as every year goes on, because I've been teaching this class, well, since 2002, so 18 years, as time goes on, right, more and more experiments, of course, are done. So I'm not really asking you to come up with something novel per se, but it will be novel in the sense that if you haven't gone into the literature and you're just thinking of it on your own, it's novel for you, okay? And that is fine. So just really kind of encouraging you to like not go looking just into the literature for ideas, but rather look at your class notes. Yeah. And just think about what you've learned about what your disease. And if diagnosis was really straightforward for your disease, it might be hard to think of a way to improve it, but maybe treatment is going to be helpful. Or maybe there is going to be something with diagnosis where microarrays could be helpful. Anyway, if you get stuck, like I said, or you want to run something by me, just shoot me a quick email if you want before Sunday. Otherwise, I'll just be looking at your drafts and I'll give you feedback then. Any other questions when you're thinking about that part?
So, Dr. Skiza, I guess I have a question about the using microarrays for diagnosis. Is it commonly used as a technique to like diagnose a disease with microarrays, or like using a more validation experiment like northern blotting or NC2 hybridization instead?
Yeah, well, that's a good question. So I think you could think about microarrays for diagnosis in maybe a couple different ways. Um, so when you think about um, like the experiment 3, figure 3 that we went over with the diffuse large B cell lymphoma, if we think of that example, because we just talked about that one, right? What they did is they started with one disease, they did microarrays, and they realized there's really 3 diseases, right, that are kind of distinct from one another. So you could imagine doing a microarray just to find out if there are distinct subcategories of your disease, right? Because that would just help with diagnosis eventually. So you could think of this doing that first microarray as the first step towards improving diagnosis, just to see if there are subcategories of your disease. Okay, that would be fine. A second thing you could propose would be that maybe you've done a microarray and you realize that there are, for the lymphoma example, that there were 3 subtypes, right? 3 subcategories of lymphoma. So then once you know that, if you have a patient, you could just compare their microarray to the 3 existing ones you already have to see which pattern they matched.
Mhm.
That would be another way to do it. A third example would be to say, okay, look, in germinal center B cell-like lymphoma, right, that first subcategory, we know that genes X, Y, and Z are all at high levels. So maybe we just need to look at genes X, Y, and Z, right, for like diagnosis. That maybe we don't need to do a full microarray for later patients. Once we've done the first microarray, right, the later diagnosis could be narrowing in on specific genes to look at the levels of expression.
Okay. Okay.
So yeah, so I'm glad you asked that, Mohammed, because hopefully that was helpful to everyone. But also it reminds me to say that in your experiments, right, in your 4 or 5 experiments, you can build on them. So if experiment 1 is, for example, a microarray to answer one kind of question, you can refer back to that microarray for experiment 2 and use it in a different way. Okay. So, because what I just was explaining was kind of 3 different scenarios, right? First doing the microarray, that's one experiment. And then the other examples were using the microarray results and then doing maybe a northern blot, or right, doing something different, or doing like a different microarray. See what I'm saying? Like the examples 2 and 3.
That's generally what I found in literature. People do like an RNA-seq or microarray just to screen for differential expression of RNAs, and then they do like a validation experiment with northern blot or western blot or some kind of like narrowing in on certain genes that they suspect. Yeah. Okay.
Exactly. So yeah, you can do those things. Did that help, Mohammed?
Yeah. Thank you. Thank you so much.
Well, feel free to like keep asking if that didn't quite answer anything. Any questions from anybody else about what we were just talking about? Any questions about anything? How we doing? All right, I'm not seeing questions. if you haven't thought about this too much yet, you may have questions. So again, feel free to email. Parting thoughts from me for today. Obviously, focus on this. This is the next, last chunk of the paper, next big assignment. And then a week from today, we're doing the peer review of sections 1 to 5. So I'll have everyone's, if you've turned it in, I'll have, you'll have gotten your revisions for, obviously you already have your revisions for parts 1 to 3. I'm working hard on trying to get the revisions for parts 4 and 5 back to people. I've already got about half the class done, so I'm getting there. I'll get another couple done today and tomorrow. So hopefully you'll have, a good amount of time to start incorporating those revisions. The more you get done before the peer review, probably, the better the peer review will be, because it'll be closer to your final version. So that's what I'm trying to say, is that if you have time after finishing the outline, which is due Sunday, then for next Thursday, try to get as many of the revisions on sections 1 to 5 done. So we're not peer reviewing this last section, just parts 1 to 5. Okay. So I will be typing up some specific instructions about how the peer review will go and how we'll do it. And there's a form you'll fill out to give feedback to one of your classmates. Okay. So any questions about the coming attractions? As always, if you have anything that comes up and you need extensions, just email, keep me in the loop. I'm working with a lot of people who just have different situations kind of going on, so that's no problem. But it would be great to get, as many of these papers wrapped up in the next 2 weeks. That will be an awesome accomplishment. Questions on where we're going? All right. Well, the sun is shining. It's cold, but hopefully you got a bit of vitamin D. I still find, even it's been so cold this past week, but and all that snow. I don't know what's going on with that. We love Michigan in April, right? If it was normal times, that's what we'd be talking about in class, is the weather, right? But anyway, try to get outside a tiny bit. I think it helps. Oh, is there a final? No final. The paper is actually worth more than the midterms, right? Overall, the paper is 28%. So no final exam. So that's a happy thing. All right. Well, have a great day, guys, and I'll see you next week.
I don't know if you wanted to know, but after graduation, I applied for this job as a MA in, my sister lives in Indiana because she's in the military. So she already lives down there. And I was like, well, I'll just apply and see what happens, because I wasn't having much success here. And they offered me a job. So I guess I'm going to go down there and switch it up a little bit from, like, right now I'm a scribe in the ER, but I'm not consistent hours, I don't think, once I've graduated, because I want more full time. And they told me they're looking for someone full time. So I guess, I don't know.
That sounds good.
Yeah, but I don't know, like, I'm more worried about the MCAT, since they just keep canceling all of the
I know.
and tomorrow they're like reopening it up to, like, I don't know if they're adding dates or what they're doing, but anyone that was already scheduled is obviously going to get the first dates to open up, so I don't know.
Yeah, I was reading more about just kind of how med schools are handling this, because of course they know that you guys can't take the MCAT.
Yeah.
And the same thing is true, for the high school kids, because so many SATs have been canceled.
Right.
So I know the med schools, some of them, and maybe it'll be almost all of them, I don't know, are saying that you can apply without your scores, that they will even review your application without scores, which they don't normally do, with the idea that, like, eventually your scores will get to them. you know, that like, even if it's in the fall, you know, like well after that initial review, they might even, you know, I don't even know if they would do interviews before you'd have MCAT scores, maybe. So, I mean, they're gonna try to, well, and they don't have a choice, but they're gonna have to work with students.
Yeah.
Yeah, because I mean, you're all in the same boat.
I would be nervous to apply without my MCAT score though, because I don't know. If I bombed it, then I'm like, oh, I just wasted all that money pretty much.
Well, I 100% agree that usually we say wait till you get those scores because it is so expensive, right? All of it, the applications and everything. And then the secondaries, they want more money, right? And a lot of the schools will just give you secondaries, like even if they don't think you're going to be super competitive, which I think is terrible, but they do it. Um, so yeah, it is a lot of money. Um, and I guess if you're doing some practice ones and if you have any guesses at where your score might be, that's probably the best you'll be able to do, you know, to decide whether to apply this cycle.
Mhm. I know. I'm already like I was supposed to apply last year and then I didn't do it because my sister got married and it really screwed up when I was going to do the MCAT and stuff, but I would hate to put it off another year because if I don't get in the first year, then I'm going to have to apply again and then I'll be 3 years out from when I
Right.
should have. So,
Yeah, I mean, one possible approach, or one like, I mean, there's only two approaches. Either you apply or you don't, I guess. But one way to think about it is like if you apply and if the MCAT score turns out to be not where you want it to be, maybe it's not a total waste of money because you can call schools, like if you apply and you don't get in, um, you can call them and ask for feedback on your entire application. So that at least would give you feedback on whether there's any other areas
okay.
they think need improving to be really competitive besides the MCAT. Like you would know obviously about the MCAT part of it, but you would at least get feedback on the other parts, and that can be valuable.
Mhm.
Um,
Okay. I didn't know that was an option. Is that just like you call the admissions of their school and
Yep, yep. And I mean, we recommend this like really strongly because, you know, a lot of, like you said, a lot of students don't get in that first try, or they get waitlisted, you know, they get close, but they don't quite get in. And so you got to find out why, you know, like what is it, how do I spend my time? I need more clinical experience. Do I need, you know, well, whatever it is, you know, a higher MCAT score. And sometimes the feedback is surprising. So it's definitely worth doing. And every school might give you different feedback, right? So if you have particular schools you're interested in attending, finding out how they're evaluating your application, right? That's what's really helpful.
Mhm. Okay. Do you have any like resources that you just already have for applicants like I don't know, examples for personal statements and stuff? I know I can like look at them online, but it's just I don't know. I'm
No,
nervous about writing that. I should have already started writing that. But
No, I think you're fine. I don't think most people have really worked too much on theirs. And that's a really hard thing to write for most people. It's a very different kind of writing than scientific writing.
I know
Um,
all college I've only ever written like lab reports and research papers and they're like, "Oh, write a story." Like, I don't know.
yeah. Yeah, it's really hard. So I do have, I'm making a note. I'm gonna send you some examples where of students who got in, so we know they worked, and they've given me permission to share them. So yeah, so I can do that. Um, and then I think I also have, I'm pretty sure, like a tip sheet, like of, you know, things to, like guidelines, like on what kind of writing this really is. I'm sure you've probably seen some of them like from online things. Um, and the other thing is I'm happy to look at it if you want. I know Amber Schneider also is happy to review things, you know, but I mean, the biggest thing is like finding some, like hopefully more than one person maybe to review what you've written, and to be prepared that, like, just like we were talking about with this paper, I think just getting something down on paper is the hardest part, like the getting it started.
Yeah.
And usually when students give me a draft, I mean, not all that many students give me drafts these days, but some students still ask me for feedback. Um, you know, usually what I see is like a lot of like a lot of different ideas, and then I can kind of help them like pick like, hey, I think this one is going to work really well, and let's expand this one, because they do want it to be like very detailed, right? In that storytelling kind of way. And what they don't want is like a series of different things that you kind of describe superficially.
Yeah.
So, but sometimes it helps just to put down on paper the different, all the different, all the different ideas you have, right? And write about them a little bit. And then you can, then it, you, we, together, like, I can help you figure out which ones that we think are going to be easiest to expand.
Okay. Yeah, that sounds like a good idea. Thanks. I
Sure,
just don't
yeah.
know, like, there's nothing like that's crazy that happened to me in my childhood that was like, you are going to be a doctor, and that's, you know what I mean? Like, there was nothing that really pushed me to it. I was just like, oh, that probably sounds like a good idea.
It does not have to be anything from your childhood. And in fact, I would say it probably is often better if it's not.
Okay.
They really want to know about some experiences relatively recent, like during college, some experiences where you've kind of been able to reflect and like, you know, gotten something out of that experience.
Mm,
Helps you know that you want to be a doctor.
okay.
Or, I mean, right, so it is a really tricky thing to write, um, because you want to answer the questions why you want to be a doctor and why you're qualified, but you can't just say, I want to be a doctor because, right? You want to answer that question like in this sort of sneaky way of like explaining it without directly answering it. It's very challenging.
Okay.
Um, but when you read some of the examples online, and I think they are really good essays, sometimes they're about like travel or like something that seems a little unrelated to medicine,
Mhm.
but is demonstrating qualities about the student that are relevant. The quality part, or personality characteristics, competencies, whatever word you want to use. So sometimes it doesn't have to be 100%, clinically clinical stories.
Yeah. Yeah, that's mostly what I'm worried about is just that my personal statement isn't gonna be interesting, and I'm just gonna be like shot out already. Like, I don't know how to stand out to other people. Like, I have all the basic things that everyone else has, I think. Like, probably a pretty average MCAT score. I'm not really sure what it's gonna be, an average GPA, and then I've done scribing, and then I'm gonna do some clinical work with this medical assistant job, but then volunteering, which is what everybody does, I think. So pretty much.
Yeah, no, I think lots of people feel exactly how you feel.
Yeah.
Excuse me. And I mean, I don't think it's realistic to think that, students are going to have like some crazy unique experience. I mean, I don't know, very, very few students are going to have that. So I don't think that's a problem. But I think it's kind of how you write about these things. Um, and so that's why, starting a little bit early. I mean, I would say April is still somewhat early because, you know, if you finish your application by the end of June,
Mhm.
I think that would be perfectly good. So that still gives you some time, couple months. Um, yeah, I don't know. I don't think it has to be um, anything you would even like. Sometimes I'm trying to explain, I don't know, I'm having a hard time with the words, but I think sometimes it's something that seemed like really mundane, like, not like a big deal, but depending on how you write about it, it can actually kind of be transformed into something, um, something, I don't want to say not special, but something interesting, you know. And I guess the other thing I've read is that it should be something you want to avoid it being generic. So that's probably why you were thinking, oh gosh, I need something special, right? Because like, oh, it has to stand out. But I think that you can think about the trying to avoid being generic as just picking some experience that you've had that nobody else could have had. And that might mean that during a volunteering experience, you had a conversation with a particular person that really made an impact on you. nobody else had that conversation. Nobody else had that interaction, or whatever it is. So as long as you're giving those details that make it, really clear and kind of bring it to life, then that's all good.
Okay. Yeah, that helps a lot. Remember
Okay, good. I felt like that was very rambly.
Amber had told us that we should have already started our personal statements back in December, and I was like, it's kind of ambitious, I think, because I have other things, I don't know. But so that's what
Yeah.
also made me worried. I was like, I'm pretty late to the game, but I don't know.
I don't think so. I don't know. I mean, I understand she doesn't, she wants students to be thinking about it early. I mean, it's not bad advice. It's just everyone has different things they're juggling. So,
For sure.
you want to make sure you're not submitting like your first draft, right? Okay, that would probably doesn't work well for most students.
Yeah.
So as long as you're giving enough time that you're gonna, be able to modify it and revise it a little bit, you're fine.
Okay, great. Okay. Well, thank you so much.
Okay,
just
welcome.
yeah,
Yeah, I'll look
Nice
for
to
those
get some
documents.
guidance because I can't go to Amber just in like a normal office hour or anything anymore. I'm just kind of I don't know.
No, I know. Well, this is, that's why I put that, well, it didn't record, so I'll have to redo it. But, just thinking about what to do, after graduation, I feel like I often have a lot of conversations with students, like one-on-one, even just at the end of class, or just, it doesn't have to be a big formal thing, but I often end up talking with a lot of students about what they're going to do next. Everyone's in a different place. And those are, I don't know, those are really important conversations. So that's another big thing we're missing by not being on campus.
Yeah, I agree with that.
Yeah. Okay.
Well, this was really helpful. Thank you.
You're welcome, Jamie. Okay. Have a good day.
Thanks, you too. Bye.
Bye-bye.